So it's a great pleasure to welcome Clara Hu to join us as today's BioSpark Innovation Talk series. And of course, we continue our recent trend by inviting more and more AI-related speakers. But of course, we are more focused on biomedical AI or AI for drug discovery. That's why we probably won't be chasing down the lane of those latest trends like cloud code type of AI, but we definitely want to eventually touch upon those topics. So before we get started, as people are familiar with BioSpark events, we probably give a quick introduction of the BioSpark. So we are basically a nonprofit organization started in 2019. And first from the greater Boston area, try to bring together different people from biotech, pharmaceutical industry, academia, and later more and more people from investment and venture capitalists join BioSpark to share information, and which is quite stimulating to exchange ideas through regular events. Initially, our main events are online through different webinars or some in-depth discussion. And we have four co-founders, Qing Hong, Teng Hui, Yan Jie, and Wang Jun. And we are later including additional committee members, including myself, Yue Hong, Li Hua, and Leon. And we basically try to expand our kind of footprint or outreach beyond just in North America. So we initially have many members, co-founders mainly from the greater Boston area or greater New York area. Then we're starting to have mainland China presence because many of our original members could go back to China to start their own companies, becoming CEOs or key leaders in the China biotech. As people realize in recent years, the hot topic is about Chinese innovations. There's a lot of innovations from the biopharmaceutical sector coming from China, which is a very good time to bring together what we have in the United States and in the mainland China, as well as some other places. You can see that we're also starting to have more members from the West Coast, anywhere from the United States, and we try to also slowly introduce more European members. And you can see in terms of the distribution of our sectors, biopharmaceutical sectors still represent 40% of our members. And we're pleased to see the investment and consulting sector now compose about 22% of BioSpark members. And then academic researchers, 20%, and we try to be more and more diversified as we go along. So we basically hosting four types of events on a regular basis. And of course, the most well-known event is the monthly Biotech News. And because we were able to maintain the monthly cadence, so since the pandemic around 2021. So every month we get together and talk about different topics. So that's kind of getting BioSpark recognized by many people in the community. And since 2025, we also changing the format of monthly news a little bit by switching to not only the slides, but also the order communication are done by English so that we can encourage non-Chinese members to join us, the discussion. Then monthly biotech news basically cover many topics, many disease areas. In case some topics need some additional deep dive, we also have the scientific series, which, for example, during the pandemic, we have multiple discussions on mRNA vaccine or LNP delivery. And in 2023, we even go so deep with the PD-1 history about the immuno-oncology. And there's a lot of those kind of topics worth some scientific series. Then we also have the leader talk, which is basically one-on-one interviews with key opinion leaders in the industry, as well as some recent published academic breakthrough. So we will invite those PIs also to have a kind of leader talk one-on-one to just share what their experience on the career development and both in the academic and industry. Then, like today's talk, we have also this innovation talk series, which is we pick only one topic, usually either one speaker or two speaker, more often is one speaker, just to try to catch up with what's the latest trend in the industry and academic research. Of course, in recent years, as people can well understand, AI, artificial intelligence, continue to be the hot topic. That's why, if people remember, if you join us in 2021, we invited Dr. Jingbo Xu to give a talk on AlphaFold 2. At that time, it just came out. Then in 2023, I remember inviting different topics like SCGPT or SCLong, just to talk about how do they apply foundation model to the single cell RNA-seq data. And today's topic is just a continuation. It's try to see the multimodal, multi-omics type of AI applications. So that's why it's very much not very regular happens. We only have like up to like 5 speakers per year, but we're hoping to have more frequent guests join us in the Innovation Talk series. And this is just a snapshot about what kind of events we have been hosting since 2021. And many of members, we probably still remember some of the talks are still very memorable. For example, we have the PD-1 history, as well as the lot of digital health innovation topics are quite interesting. So this is just a snapshot of the most 2 recent episodes of Monthly Biotech News, and both in February and March. Our next one will be talking about the April news, will happen on May 2nd, just as a preview. May 2nd is a Saturday, so we welcome people to join us again to talk about those areas. So today, I'd like to set the stage for our guest speaker, Clara, in terms of this is just our disclaimer in terms of all the opinions and just expressed in any of the webinars are just representing our guest opinion. Does not represent the BioSpark, especially when we talk about biomedical and anything related to medicine. People just should treat this as each individual opinion, not BioSpark's in terms of position. So in terms of setting the stage up for Clara's talk, and I just remind people just the reason for the booming of the recent years artificial intelligence is mainly because we are moving beyond the supervised learning. If you remember the history of AlphaFold, initially AlphaFold 2's success is still pretty much on supervised learning. But as people realize, other than protein structure, protein data bank, there's not easy to get labeled data. So because so large volume of the data is unlabeled, so that's why ChatGPT becomes successful, because natural language from internet, a lot of those data are not labeled. Then the Google's transformer leading to the innovation called GPT, which is called generative pre-trained transformer, which can train the whole text, natural language text in an unsupervised or self-supervised manner. So that created a lot of additional application. So that's why probably people want to just on the high level understanding what's the difference between the traditional supervised learning, which requires label data, and the self-supervised learning is more like the natural language. If you can mask certain words in a sentence and ask the model to predict those missing words by having a large volume of natural language data, usually they can do a better job and create something called embedding, which Clara will go in a little bit deeper. And then, of course, there's always the semi-supervised learning. So combining some of the label data with the larger majority still unlabeled data and provide together, which enables something called transfer learning, which is a very important topic. If you people knowing Eli Lilly's TuneLab or federated learning, a lot of the federated learning is just kind of motivated by the idea of transfer learning. Then this is just to emphasize more unsupervised learning, which will be Clara's main topic, is because a lot of data is unlabeled. So Clara came from UCSD Adler Lab. Their previous publication was using supervised learning. So by the time Clara joined the Adler Lab, they realized a lot of data we can treat it as an unsupervised learning framework, which leading to some very interesting innovation. But one concept I don't want people to get lost. People hear about those terminology very often. Probably Clara won't have time to go too deep in terms of the introduction. I just setting the stage for her in terms of what is autoencoder. Why people keep hearing the concept autoencoder. I just want to demystify. There's nothing mysterious about autoencoder. essentially you can think about as an extension of PCA, principal component analysis. So what principal component analysis is so well known since 1933, it's basically dimension reduction. So it allows you to treating the noise data by reduced representation. For example, denoising. If you have a handwriting 6, if you're taking a photo, it's kind of blurry. So how do you remove the blur? How do you make the picture a little bit clear? So the one very important technique is just to reduce the dimension. You reduce the dimension so that it can only focus on the essential features. Then once you reconstruct the image using this reduced representation, a lot of those noisy, unimportant features will disappear or being hidden so that you get a relatively clear picture of the handwriting. So to really extend PCA, because PCA is limited, it's a linear method, a linear dimensionality reduction method. So by introducing the neural network or deep neural network, which is the kind of current AI revolution came from. So those deep neural network, you have multiple layers, and each layer can do nonlinear transformation. So that it's very flexible. It allows transfer learning, whereas PCA cannot do transfer learning. PCA, every time you get a new data set, you have to rerun the PCA like a gigantic matrix. But with encoder and decoder setting, you have this autoencoder. And the key thing I want people to take away is that it's not the input or output that's important. For autoencoder, it's always the hidden layer, in the middle. What's in the middle is the important, because the output is just used to calibrate the model. But what's in the middle is something called a bottleneck layer. The bottleneck layer captures the most important information. So if you want to think about what's most important thing, it's not output, because output is used to calibrate the model. What's in the middle is really essential. So Clara's talk will illustrate that point even further. So I just want to give people some example of application of autoencoder. It sounds like a very simple idea, but it's really useful. For single cell RNA-seq, for example, people are familiar, there's dropout issue. There's certain genes, because the library prep, because the single cell nature, it will have dropouts. So that's not a biological zero. So some genes are not expressed. That's called structure zero. That's really unexpressed gene. But a lot of genes are expressed, but due to the methodology of the single cell technique, there can be some dropouts. To address those dropouts like missing data, there's a way to use autoencoder illustrated in this book. People are interested. Then this kind of model can distinguish what's the dropout, what's structure zero. So that's one very simple implication. It's similar to the denoising of the image I illustrated in the previous slide. Then, of course, Clara's work in the ADC lab was motivated by having different data types, different morphology data. But in a 2022 publication, people in the field already tried to integrate spatial transcriptomics data with the imaging morphological data. That's always one of the most important features of bioinformatics or computational biology is when you have omics data, essentially it's a measuring of gene expression, protein expression, or metabolite abundance. And that usually does not come with spatial temporal information, whereas cell-based imaging or live animal imaging contain the spatial temporal information. If we can bring omics abundance data with the imaging data together, that integration is always very important to people familiar with computational biology. So what Clara's work is basically moving beyond the transcripts, a protein. Not only that, she studied protein-protein interaction. So she can bring the protein-protein interaction together with the protein localization together in a very coherent network so that they can make some very interesting model building. So let me just quickly introduce Clara's background. And she got her bachelor's degree from the University of Wisconsin. And from 2015 to 2019, then between 2019 and 2021, she got her master's degree in biotechnology from Brown University. And then in 2021, she moved to UCSD to become a PhD student and join Trey Adler's lab, a very famous lab in the UCSD. And Trey was very famous for systems biology, working with Lee Hood before 2001. And during her PhD work, she was able to apply large language model for the annotation of those different pathways or modules, and as well as the work she will present today. So as of part of that, as a kind of note to that success, she was voted the 2026 Forbes 30 under 30 in the science category. So let's welcome Clara to BioSpark Innovation Talk. I will stop sharing here, and Clara, if you're ready, you can start to share your screen.
Yes, and thanks, Ethan, for the warm introduction, and also thanks for kind of covering a lot of the background of the technical part of the study. And yeah, I really appreciate it. And I really appreciate to having me here and excited to share my work. Yeah, my name is Clara, and I recently earned my PhD from UCSD, and I am currently a postdoc scientist at J&J. But today's talk is mainly focused on my work during PhD, and the talk is titled as Multimodal Mapping as a Foundation for Virtual Cell. So this is a picture of a cell. I believe all of you are familiar with it. Every tissue in your body is made up of billions of these cell units. And when you look inside, you can see the level of complexity inside the cell. And these are thousands of different types of molecules that work together inside the cell, like factories that never stop, and they keep the cell alive and function. So we know that the cell is organized in a hierarchical manner. The bottom of this hierarchy is as there are individual proteins, thousands of them. And when you are building a Lego model, the basic pieces we are talking about here is proteins. And proteins organize together to form protein complexes and function as as pathways, and then further housed into bigger organelles, and all of which together make up the cell as a whole. And what's remarkable is the range of scale here inside the cell. We are talking about structure that span 3 orders of magnitude, from individual proteins that are around 5 nanometers all the way up to the cell itself in microns. So here's what I kept thinking about. What if we can navigate the cell like the way we navigate the city? Thinking about Google Map, you can zoom in and out, and zooming out, when you zooming out, you can see the whole city, the major districts, the highways, and then you can zooming in on the Google Map, see specific neighborhoods, see individual street and buildings. And you can click on those restaurants to see the complete profile of those restaurants, the reviews. Now imagine, what if we can have the same thing for the cell? You can zoom out, see those major organelles, zooming in to specific protein complexes. And you can click on those protein complexes and learn about what it does. What are the compositions, and what happens when things goes wrong in disease. And this is what I mean here by a virtual cell, a data-driven navigable model of the cell where any researchers can ask a question: where does this protein located, and what are the protein community do? What happens when things break? So as scientists, we've been studying this cell for decades, but we know a lot about individual genes, individual proteins. But the truth is, much of the cell's internal structure and subcellular function remain uncharted. We still don't have a good map of how everything fits together. You may ask, well, why do I care about what things going on, or how things are organized inside of the cell? So what if I tell you that you only see 10% of druggable biology because the other 90% of the cell is still not mapped? Then this become a big bottleneck in drug discovery. This is exactly why I want to close the gap. And my work was centered on this one question: can we use multimodal data and AI to elucidate what is inside of the cell? For today's topic, I'm splitting the story into two sections. Section one is about building this navigation system for the cell map. And section two is about interpretations, using large language models to understand what the map can tell us. And together, these two stories form a framework I think of as a foundation of a virtual cell. So let's start with the map. To build a map of a cell, we first need the data. Instead of relying on single type of data, I collaborate with scientists to bring together two complementary modalities in U2OS cell at very large scale. This gives us very different and complementary perspectives on protein localization. Let me explain. So the first technology is called immunofluorescence images, or IF. In IF experiments, individual proteins are tagged with fluorescence antibodies. Then we take confocal microscopy images of the cell, see what exactly are those proteins live in the cell. It gives us a very global view of where protein localize inside of the cell. Here I'm giving you 4 examples of how those images look like. The green color is the proteins, and these proteins show clear localization information in nucleoli, Golgi apparatus, cytosol, and nucleoplasm, if you see the green dots. In total, we collaborate with Dr. Emma Lomberg's team from Stanford, and together we collected over 20,000 images of proteins in the U2OS cells. And the second modality is affinity purification mass spectrometry, or APMS. In APMS, a protein of interest is called BAIT, and it pulls down the interacting partners called praise. Then use the mass spectrometry to identify those proteins. This gives you a local information of which proteins are physically interacting with one another and gives you the neighborhood information of each protein. Our collaborators from Harvard Medical School generate over 36,000 protein-protein interactions from APMS in the U2OS cell line. So we have this massive interactome as our second layer of data. So these two data modalities captures opposite ends of the spatial spectrum. The images kind of gives you this global view of the broad compartment where proteins are located. Interactions gives you the local view, who's in which neighborhood, who's in the community, who's in this community, and how they talk to each other. Integrating those data gives you a much richer picture than either alone. It can inform the makeup of the protein complexes inside the cell across scales. So now I want to go into how do I actually combine these two very different type of data, because images are a matrix of pixels and interaction network is a graph. They naturally don't speak the same language. So first, I obtain the protein embeddings from each single data modality using two separate neural networks, DenseNet and Node2Vec. So embeddings is a common term in machine learning describing a low-dimensional representation that is extracted from high-dimensional data. For example, here, each protein is then represented by a vector storing the location information that was informed from images and the neighborhood information from the interactions. And this step ensures that they are now in the same dimension and they are represented by those by vector space. Then I train an unsupervised contrastive learning model to learn the protein position representation. And thanks for Ethan for explaining the unsupervised machine learning. So it basically places those proteins information into the same unified space. Here is showing the space with over 5,000 proteins. Each dot here is one protein, and the position is defined based on the integrated information from images and the protein interactions. Now let's dive into the model itself a little bit. So this machine learning model is trained to minimize two main loss functions. The first one is called contrastive loss. So the idea is we sample the protein triplet, an anchor protein, a positive protein, that is the protein that is similar to the anchor, the negative protein, the protein that is dissimilar from the anchor. And the model is trying to learn to minimize the distance, so pull in the positive and to maximize the distance to the negative, so push out to the dissimilar proteins. This step learns the geometry of the cell from the data itself without really relying on those non-standard hand-labeled annotations, or also it works with things that does not have annotations at all. So we also add a reconstruction loss. So in the model, we have a decoder that tries to recover the original embedding from this unified space. This ensures that we don't lose any information during the integration step and still recovers the original data. So combining the two loss ensures ensures the model have informative and faithful to the data. So for those of you who don't know much about machine learning or computational modeling, in simple terms, this method allows proteins that are closer to each other to be close in the unified space, and the ones that are further apart will be pushed away in the final unified space. So we essentially learn the spatial logic of the cell. Now we have placed the proteins in their location based on the integrated data. We can then connect the dots based on their spatial proximity and create this huge connected network of over 5,000 proteins. If we take a snapshot of this large hairball, it represents all the interactions happening in the cell. Or we can say it is the topmost node of the hierarchy. By looking at this cloud of interactions, you can roughly see there are some components with proteins that are tighter to each other. So if we dial the focus a little bit, we can then see that, oh, here is a component that can represent, that is the cytosol. And we can we can dial a little bit more, look closer into the cluster of proteins that represent the endomembrane and moving down the layer of the hierarchy. And then we look at a really tight protein network that representing members inside of the endoplasmic reticulum that is going even down one more layer in the hierarchy. So this the step that I just explained is called pan-resolution community detection. It is it iteratively adjusts the resolution, like you adjust the zooming on your camera to zooming in and out, and it reveals the protein communities at different scales simultaneously. So here is what we get out of it, this U2OS cell map. Each nested cell here represents a protein assembly. So here, a protein assembly means the group of proteins that are closest to each other from the original data. And the nesting shows containment relationship. A smaller assembly is contained within a larger one. Just like you have neighborhoods within a bigger district. So the outermost circle is the cell. And as you zooming in, you can see the major compartment, those bigger ones, the nucleus, the cytosol, and you can go into deep depth of how you can see the nucleoplasm, and even you can see even further detailed of smaller assemblies. Here I'm just showing one layer of resolution. But in total, we resolved 275 protein assemblies, including over 5,000 proteins. This is the largest hierarchical map of a human cell ever built. So, and here is another view of the same hierarchical relationship in tree structure. You will see this later in the slides, so I want to present it up front. It contains the same information. You look from the top to bottom, you move, you can see more detailed organization as you're moving down the tree. So you can see there are two different colors of the node. It represented that over about half of these assemblies matches with well-known assemblies in reference databases like gene ontology, quorum complexes. So this means that we recover known knowledge, which is a good validation. But also you can see there are 142 assemblies that are in purple, and they are not in any, meaning that they are not in any existing databases. They are genuinely new, and that's where things getting really exciting to biologists, because we are discovering new things about cell biology. And I will come back to these nodes later. So for this map, we also translate the size of each assembly to an estimated physical diameters. We found that they span 3 orders of magnitude, from nanometers to small to microns for the cell. So if you remember the scale that I showed in earlier slides, those represent the true scales of the cell. So meaning that the map is truly multi-scale and represent the actual scale of the cell biology. This is really an aha moment when we find out this, when we plot find out the physical diameters. Then we build an interactive web portal so that any biologist or any person without coding background can explore the cell map, just like Google Map. You can zoom in and out, and to search the proteins. You can search the proteins, or you can click on the assemblies and see what is underneath it. And when you click on the assembly, it will show the interactions between those proteins. And when you click on each proteins, it will then show the underlying imaging data. So we want to build this model, to build this map as a community resource for for people working in cell biology, molecular biology, or all kinds of biologists that wanted to see understand the protein and how it connected to different compartment and different members. And yeah, so now let me tell you what I can actually do with these maps. So we are not just stopping by presenting a model. We really did explore 5 different downstream biological applications, including new insight in 3D structure, assigning new protein functions, compare the changes in terms of interactions between cell types, track the multi-localized proteins or complexes, and also using the map to assist interpretation of cancer mutations. I will focus on 3 today because of time, but I'm happy to discuss the others during Q&A or offline. So first story is about 3D structure modeling application. Here is the RAG regulator. It is a well-characterized protein complex that regulates a really important signaling pathway called mTORC1, which controls cell growth and is a major drug target in cancer, actually. The 3D structure of RAG regulator has been already resolved and is presented in PDB, the protein database, protein structure database. But when we looked at our cell map, our computationally inferred cell map, the integrated data clearly placed two new members, ITPA and BROX6, in close proximity to the known RAG regulator members. So our multimodal data is suggesting that these model might be new members of this complex. So I collaborate with structural biologists from UCSF to reveal this new complex structure using integrative modeling simulation, adding those two members, the ITPA and BROX6, to the existing protein complex structure. So this is the case where our cell map can help expanding our knowledge of protein complex structure. So second application is probably my favorite. This is a story about how a data-driven map led us to an unexpected biological discovery. So here is a story about DPP9. DPP9 is a serine protease. And what our cell map shows was that DPP9 was consistently placed right next to three key interferon signaling proteins, STAT1, STAT2, and IRF9. So what we know about these three proteins is that they form a well-characterized transcription factor complex called ISGF3 complex, which drives interferon stimulation gene expression. ISGF3 is central to antiviral immune response as well. But here's the thing, DPP9's role in interferon signaling has never been studied. We had a prediction from our data, but we need to test it. So we go to the wet lab and treated the cell with DPP9 inhibitors and measure the expression of interferon stimulating gene downstream from ISGF3. The qPCR result on the left-hand side is clear that inhibiting DPP9, you can see from the pink diamond, inhibiting DPP9 significantly upregulates interferon stimulating gene expression. This means that DPP9 is acting as a negative regulator of the ISGF3 pathway. So what we found is that when DPP9 is activated, it suppresses the interferon signaling. So when you inhibit the DPP9, you unleash that signaling. This is a completely new function of this protein and new indication for DPP9 inhibitors. This is exactly the kind of discovery we want to see from the virtual cell that the virtual cell map can enable. It helps complete the functional annotation of human proteome and also opens up a really exciting new avenue of potential target related to interferon signaling. So now I want to move on to the third application that brings us to oncology, specifically in pediatric cancer. One of the greatest challenge in cancer genomics is that the mutations are sparse. What does I mean here? If you look at the mutation landscape of tumor on the right-hand side, most of the individual genes are mutated in only a small fraction of patients. So here you can see those lines, but all this long tail are the genes that are rarely mutated. And often they are less than 10% of the patient cohort. This makes it very hard to identify the driver genes from the bystanders. So just imagine this: if you are looking at individual mutated genes, it will look like those dimmed light in the cell, and hard to tell the bigger effect or the role they play in cancer progression. But here is the power of having a systematic map. Instead of looking at the individual genes, we can ask which protein assembly or which functional systems are being hit by the mutation recurrently across the patients. Even if no single genes are mutated across the cohort, the same assembly might be disrupted again and again through different genes, just like what I'm showing here. Now you can see that those are the units that always being hit. And this is the concept of convergence in cancer. The tumor can find different paths to disrupt the same functional unit. So having that idea, I used a pan pediatric cancer genomics data from a large Nature study published in 2018, applied a regression model to aggregate cancer mutations onto our defined cell map. We identified 21 protein assemblies that are recurrently disrupted in pediatric tumor. These include like transcription regulations, chromatin remodelings, and collagen biosynthesis, etc. And those might be potential cancer drivers. So within these 21 assemblies, there are 250 mutated genes from the tumor samples. Then I validated these mutations against independent mouse transposon screening dataset. It is a large-scale in vivo screening designed to identify cancer driver genes. So from those 250, 102 are validated from the screening. And of those validated proteins, 63 of them are completely novel pediatric cancer drivers identified the first time in this study, and 10 of them are known adult cancer genes, but haven't been associated with pediatric cancer before. So those together are potential novel drivers to follow up in the context of pediatric cancer. This guided the cancer research to a narrower searching space. Now I wanted to zoom in on one of the highlighted assembly. We call it an ENCORE associated transcription regulation complex. We found that multiple members had been independently identified as cancer drivers from the mouse screening. This gives us higher confidence that this assembly is genuinely important for cancer progression, especially in pediatric cancer, and gives cancer researchers a specific and actionable set of genes to follow up with. So I want to just summarize this, just kind of wrapping up the section 1. I've shown that we build a largest comprehensive multi-scale protein map of U2OS cell. We integrated in imaging and protein-protein interaction data using the self-supervised machine learning model. We resolved 275 protein assemblies spanning 3 orders of magnitude. And we demonstrated that this cell map can enable different biological applications, from identifying new protein complexes, complex structures, assigning new functions, as well as interpreting the pediatric cancer. Now here's the story taking interesting turns. Once we built the map, we realized that we had a new problem. You probably remember I mentioned this. There are over half of the nodes here, over half of those protein assemblies are did not match with any of the existing databases. Those are genuinely new findings. And the bottleneck here is how can we interpret those if they are not in the existing knowledge? So now the bottleneck is no longer the data or the computational method, it's more on the interpretation side. So this bring sorry, yeah. So this brings me into my next story today is to interpret the subcellular functions with large language models. So just beside building the cell map, just generally in omics research, we identify gene groups that are function towards the same biological condition or disease, but biology needs interpretations. Traditionally, biologists go into the literature, synthesizing the functions, and write the comprehensive report. These process can take days, even weeks or months for a single gene set. This is hard to scale up. And the question I asked back then is that, can I use large language model to act as those biological interpreters, or can we use AI to read the cell map that we just built? My first instinct, like many of you might have had, is to just ask ChatGPT. Well, back then is when ChatGPT just came into the world. So I gave it a list of proteins and said, like, tell me about the function of this gene list. Honestly, it gives pretty good result. The initial result, it looks pretty impressive. It gives me a whole page of text explaining the function of those genes, how they linked to each other, and propose the name of the gene set. But when I read it carefully, I saw some problems. First of all, the proposed name was like really broad and meaningless. A cellular regulation and homeostasis complex. What does that mean? It's basically everything inside of a cell is like that. So there's no way, and there's no way, but it is because I have some background knowledge about those biology, and so there's no way to know which part of the response from LLM we can trust. And you probably already know that AI is really good at giving you confidently wrong answer. In biology, these confidently wrong answer is even worse than no answer at all. So my attempt here is my takeaway here is that first the naive prompting is not gonna work. I need more sophisticated engineered prompting guidance to the LLMs, and also in robust evaluation to assess the output from the LLM. So first, I built a automated pipeline for LLM-based gene set functional analysis. This pipeline used a carefully engineered prompt to guide the model through a structured reasoning process, ask it to analyze each gene, identify the coherent theme between those genes, and propose a specific name for the LLM, assign the confidence score for this gene set and the name. I applied the strategy called chain of thought prompting back then to guide the LLM step by step. But this was the work that was developed 3 years ago, and modern LLMs right now already came with this thinking process internally. So I'm not going to go into super detail of how I engineer the prompt and the technical part of how to build this pipeline. But I really want to point out here is that in the prompt, I asked it for a confidence score. This is the step. It's not just the confidence score itself, but the idea of asking it to self-reflect on how much the proposed annotation is accurately assigned to the functional gene to give us a hint of how much we can trust the result. And more importantly, I ask the model to teach it to just say no, asking it to respond systems of unrelated proteins when it figures that those genes should not work together and there's no coherent theme between those genes. Because in real life, there might be artifacts, contaminations, false discoveries from experimentation or your downstream analysis. So it is really helpful if we can get a hint of potential noise in the data or in the analysis process. So as expected, the result looks very good. Here's this one example when I query a 6-protein complex from our cell map, and just in second, the LLM returns a pretty good report, gives a name, ubiquitin-mediated proteolysis cell cycle regulation, and it goes through a detailed analysis assay, links all these 6 proteins through their shared role. And what would take a human scientist days or more to research on and to write, to synthesize, this system now can produce in second. And I think now it's not a surprise that LLM can do all the things in second in scale at scale. So we then wrapped this pipeline into a web portal called GSAI, allowing scientists without an encoding background can just query their own gene set of interest and get instant LLM generated annotations with the confidence score to just make it available as a community tool. So I know that all the things I've been talking about earlier, that the LLMs, the automated pipeline is kind of dated because of the nature in AI, that AI just developed like a rocket. But what I wanted to spend the most time on and bring your attention to is this question: how we can evaluate LLMs and how much we can trust its answer, and how do we develop domain-specific evaluation frameworks. This might sound simple, but it is really not. There's no universal benchmark for guiding how can we know AI understand biology. So it's still an ongoing contribution for developing benchmarks and evaluations for AI. So my first evaluation task is to ask, can LLM recover known biology? We use gene ontology as the gold standard. It's a database of gene set with human curated functional names. So we gave the same gene set to 5 LLMs and ask them to propose names, and then measure how close the names matches with the annotation from gene ontology. Now, I wanted to be upfront about things. These models, the GPT-4, Gemini Pro, Mistral, Llama 2, were the landscape 3 years ago, but right now it's kind of outdated. But if I ran this benchmark today, these numbers might look different. But this evaluation framework itself that I'm presenting is completely model agnostic. It can be applied for any type of model, even the novel, even the most modern ones. So I used the metric called semantic similarity. This metric evaluated the conceptual closeness between 2 words or phrases, not just the exact term match. The results shows that most models, including GPT-4, Gemini Pro, GPT 3.5, Mixed Instruct, kind of behave similar comparatively. They can recover known gene ontology terms. Llama 2 performs significantly worse. So the takeaway here is that most modern LLMs can identify biological functions that are in well-characterized gene set and knows about the biology. But that's is kind of simple task. The harder and more important question here is that can the LLM tell the difference between a real coherent gene set and a random one. Because in biology, false discovery, false positive are really expensive. A confidently wrong annotation can send a research team down to a fruitless path for months, even year. So we created this negative control by randomly shuffling the genes across gene ontology terms, contaminating the gene set and also contaminating the gene set, providing the gene set with just random non-coherent functions. Ask each LLM to assess its confidence. So at that time, GPT-4 refused to name a random gene set for almost 90% of the time. The other models, especially Llama 2, are dramatically overconfident. They happily generate confidently sound names to those gene set and kind of hallucinate the functional relationship between proteins and genes that don't even exist. So this is a critical finding for anyone who are in the field of AI for science. That is knowing that they say I don't know. You can probably feel that when you talk to your chatbot, any type of chatbot, they always agree with what you are saying and giving you things that sound really plausible. So our benchmark here and just giving the idea of we need to put something that are negative control to see how well it responds to random, to how it responds to just untrue to data. And here, at that time, GPT-4 did the best with our carefully engineered prompt and the benchmark. So we also compare this approach with the standard statistical approach called functional enrichment analysis. That is the test of comparing how much your gene set significantly overlap with known pathways in reference databases. GPT-4 performs comparable with the functional enrichment in terms of distinguishing real gene set from the random ones, but also it refused to name a few that are contaminated. Whereas for statistical approach, when there's enough genes that matches with the reference, it assigns the significance for the contaminated ones. Next, I want to see how these models perform in real life research scenario. I ran the experiments to analyze omics gene set by taking a pool of 300 gene clusters from different experiments, including perturbation screening, differential expression, protein interaction experiments. This allows us to understand the practicality of using LLMs in actual research setting. So GPT-4 proposed names with confidence for over 135 cases. And it can also synthesize knowledge and generate functional hypotheses about those gene set, especially for those that don't overlap with existing databases. And we looked at how the GPT-4 proposed name compared with the functional enrichment names. And again, those functional enrichment are matching the queried gene set with the reference gene set and annotated with the name from the referenced gene set. As you see from the left, in most of the cases, the name from GPT-4 and the enriched name are similar, shown in red, but GPT-4 generates a name that covers more of the genes in the cluster than the top enriched ones. This means that LLM-based names are more inclusive. Showing one example here, the top enriched term for this gene set is the negative regulation of pancreatic juice secretion, and the second top is protein ubiquitination. Combining them together, the functional enrichment covers 8 genes in their annotation, whereas the gene GPT-4's name in the underlying analysis report, it reasons out all 16 genes. So GPT-4 reasoning is reasoning from broader biological knowledge, identify the theme that integrates all the genes more holistically. LLM are not, but what I want to emphasize here is that LLMs are not really replacing the statistical enrichment analysis. They are being complementing. It covers the unseen part of the databases and covers the part that's not well documented and providing the knowledge for proteins that are not assigned to a gene set, a gene set that in the database. So here I want to wrap up and summarize the section 2. Basically, I build a automated pipeline for using LLMs to annotate gene set and run gene set analysis. And I evaluated the ability of those LLMs in terms of how much it recapitulates known knowledge and how it self-reflects, and applies the LLMs on those real-life scenarios, and noting that I used the pipeline for naming those novel assemblies identified in CellMap built earlier. So I know that the technology is dated, but the domain-specific evaluation framework remains as a bottleneck in current AI for science field that I wanted to bring everyone's attention, that it still needs a lot of scientific contribution to really develop those benchmarks, guidelines for the AI for us to really understand how much we can trust the response from the AI, how we can evaluate its result, and how we can better use it for research cases, for drug discoveries. And we really wanted to use AI to speed up the research and biomedical field. Okay, let me just zoom all the way out now to just tell you about how those two parts of the story together really means. So the cell map tells us where those proteins are, where things are, how they organize together. The LLM cell systems then tell us what those protein assemblies do. Putting them together, you have something genuinely new. It's a computation, something that you can computable. It is computable, interpretable model of a cell. That is what I meant virtual cell here. It's not a physical simulation of the cell, but a data-driven knowledge graph where any researcher can locate where proteins are in the cell, understanding its functional context, and get AI-generated hypotheses on the fly, knowing their roles in health, in disease. And I also wanted to say that there are a lot of exciting directions from the work that I just presented. For example, the current cell map that I presented is a static snapshot. Proteins in real life are dynamic, they move, and protein complexes can assemble and deassemble in response to drug treatments in disease. Building a dynamic cell map can capture those changes, and that is a major next frontier. And that's also the ongoing work that was being done. That is doing in the IDECR lab. We are also extending the work to other cell lines and tissue types. And because biological data are so rich, we of course are continuing developing better computational models to incorporate more data modalities to encompass more data, uh, imputing the missing data to build more comprehensive cell map. And on the AI side, I think the exciting direction is more deeper human AI collaboration, the automating the lab work where AI can propose a mechanistic hypothesis and human design experiment, do the experiment, then test them, really do an iterative loop, giving feedback to the AI. And what I value the most and currently continually working on is see how AI can generate hypotheses, and I am contributing to building those benchmark systems to evaluate how well those hypotheses are from the AI. And from there, before I close, I really want to say thank you to people who contributed to the two work that two story that I just presented. Specifically, my PhD advisor, Dr. Trey Edgar, he kind of guided me to through the correct direction to make all the both work success and into publication. And I want to thank all the collaborators from different institute and all the members in the IDACR lab and my funding sources and thank you all for listening, and I'm happy to take any questions. Yeah.
Yeah, that's great. Very exciting talk. So yeah, we do have a question. So how about we doing this? First, we will ask the technical question, more detailed technical questions. Then I will uh, probably follow up with some big picture, like especially your final slide, looking forward in terms of what's next, those type of questions. So one very technical question from our audience is from NZ. He's asking, how are the positive and negative proteins defined when you show the calculation of the contrastive loss function? What are the pulling? What are the compelling? What's the definition of those positive and negative? Yeah, the pull and the push.
Yeah. That's a really good question. Yeah, I didn't go into that detail, but for individual data modality, we did there is an initial clustering from the data. So we did an clustering based on how close the proteins are from individual data type. Then that's how we identify the proteins that are similar to each other. Those are the proteins that are in the same clusters, and the protein that are dissimilar are protein that's in different or distant clusters. Yeah.
So just clarify, this cluster is more defined by protein-protein interaction, not by the
No,
immunofluorescence.
it's from individual. So both data models, so both imaging and interactions, we already have the protein embeddings that
Hmm.
contain the information from image and the interaction network. And from there, we run the clustering based on these input protein embeddings to identify
Mm-hmm.
proteins that are closer to each other in individual
Mm-hmm.
data modalities.
Mm-hmm.
And that's how we define the positives and negatives.
Yeah. Yeah, one quick follow-up to this question. This is really interesting. So some proteins can be mobile. They may be
Mhm.
localized, for example, in mitochondria at one particular context, but they may originally coming from cytosol. There's the protein transport, protein post-translationally have to move to the mitochondria. So of course, you are just taking a static picture, but some protein can show up in multiple locations. Among all the data you gather, have you seen any example of certain proteins show up in different complex in different locations?
Yeah, that is really a good question. And we did explore that in the paper. That is one of our downstream biological applications we see that we saw the the integration method has still conserves the proteins that are localized in multiple compartments. For example, here I don't have the exact name of the protein, unfortunately, but we did see proteins that are located in both nucleus and in cytosol, and as well as in terms of complexes, there are same complex that are located in several compartments. We can capture those, but yeah, but I want to acknowledge that we cannot. The current limitation is that we cannot see the trajectory of if one protein was moved from one
Mhm.
place to the other because this is just one static map. But for future work on building different maps at different um, conditions
Mhm.
can potentially figure can potentially find those
Yeah,
relationships.
yeah, yeah. That's what. Then, if you can go to that slide to show the ISGF3 complex, where the DPP9 was
Yeah.
showing the negative regulation of IRF. Yeah, this slide. One thing got me curious because, yeah, you did the inhibitor experiment, which is pretty good evidence. But because we know DPP9 is a serine protease, have your team ever thought about just testing the simple hypothesis called the all-comes razor, right? Because can DPP9 cleave any of those three proteins, IF9, STAT1, or STAT2? Have you ever tried a simple cleavage experiment or any literature evidence showing could be just cleaving to downregulate?
Yeah, that is actually, yeah, that's really good approach. I guess that's also something intuitive that we think, oh, because it's a protease that we can find any motif from
Yeah.
any of those proteins if it can cleave. Yeah, we did think about that, and that it is in one of our experimental plan when we design what validation we want to test, but there are some technical uh, difficulties on how much the wet lab can do. So did
Mm.
we choose a different route, but that is really a good point of just testing if it cleaves any of those proteins. Yeah,
Yeah, because
that's
it's a new
something that.
discovery. Especially you may
Mhm.
find an immunologist kind of collaborator. They may be very interested in DPP9. Then
Right.
you can do some biochemical assays. That'll be a major discovery for down the road.
Yeah, that
Yeah.
exactly. Yeah.
Hmm. Yeah. Okay. So also, when I think about your pancreatic cancer application, you talk about discover those mutations. But one thing, have you tried because DeepMind have those alpha missense or alpha genome, try to see
Hmm.
what's the consequence of those mutation. Have you thought about bringing those information together into your model?
Yeah, that is a really good idea. Um, so when we do the do this run this as application, the alpha genome haven't been developed yet or haven't been available. But that is really a interesting idea of including genomics uh, mutations in using genome into the map.
Yeah.
And yeah, I think that's something we can
You
think
must have
about
some,
bringing.
yeah, missense single point mutation. Could be simple amino acid missense
Right,
mutation. Then alpha missense you can also try in terms of
right, right,
the stress
right. I think
consequence.
yeah, it can be used for like predicting new
Hmm.
mutations
Yeah,
and their function. Yeah, that is a good point, and that's something I can bring up
could be follow up. Hmm.
to the lab. Yeah.
Yeah, yeah. So then to your second part of your talk, when you use LLM to annotate the gene function or gene cluster function, and even for GPT-4, the best performed LLM, have you seen some very obvious example of hallucination? I know your conclusion is GPT-4 hallucinate the least among the model you tested, but have you ever still seen some very bizarre hallucination? So what would be hallucination look like in those kind of functional annotation?
So yeah, so one type of hallucination is things that I tried to oops things I've tried here, like giving a random set and see if it hallucinate any functions to those those gene groups, and that is one type of hallucination. And also we look at the assay. So because the result we have is not only the name, but the a kind of a report of how those what are the function of those genes and how they related. So one thing we did is that we have a second follow-up pipeline to find literatures from PubMed, to extract literature from PubMed and matching the text with the claims that they made in the text to kind of further to that's the automated way to validate some of the sentence, some phrases that
Mm-hmm.
it described in the text. And also we did some heavy human evaluation to the text. We did find that it sometimes they claim something that reported in any of the paper or any of the things that we can find in online. And it's we did categorize. So above, if I remember correctly, above 80% of time it's correct information from the subset that we human evaluated, and the rest of this is either in a wrong. It's something that's towards a correct direction. It's more on it's hypothesizing things that, oh,
Mm-hmm.
this is the reason that why this gene is involved in the system. So the
I see.
current it's more of the current role is true, but it said something that speculates the potential role, and that we we still count we still count those as not true information, but those are interesting because they kind of they show the case of how LM can help with hypothesize new biology. And and then that we find some cases where it's just completely missing the
Mm,
function of the
got it.
gene, gives a wrong annotation.
Next question. Then, of course, I saw many papers in the field along with this line. You are using GPT-4 and other LLM to annotate a function. But before you did this work, many people already using the LLM to annotate single cell RNA-seq cell cluster because there's some
Mhm.
similarity. When you see a lot of cell clusters, so I'm pretty sure you must read those literature, at least from your impression. How's the performance of those GPT model or whatever LLMs in cell type annotation for single cell data? Do you think they are doing a good job? Or compared to what your experience with gene annotation, how does that compare in terms of application of LLM?
Yeah, that's yeah that so first of all, I think it's more of a it's not like they do things before us. It's uh there are a lot of effort in automating the single cell annotation. I want to just acknowledge that I don't have a lot of expertise experience with a single cell annotation, but from my understanding, it was based on a lot of marker genes and
Mm-hmm.
uh, and and the LLMs can be used for identifying those marker genes and then associate the annotation to those single cell types. Um, so I think those are inherently different tasks than the gene set annotation. So I I don't I can't tell if it's there is a um, direct comparison between how the performance wise how it performs better in uh, one area than the other. But yeah, but
Yeah.
I think yeah, go ahead.
Yeah. Just quick follow-up. You can see the slide you show here, those annotation does not including cell type because some annotation, gene ontology, I forgot. Gene ontology could give you some kind of keywords in referring to cell type. Have you thought about that? Those information also, each gene have something called body atlas in a GTEx. So every gene have an expression pattern across all the normal tissues or even cancers. Those information, do you think that can bring together for LLM to digest or to help you doing the annotation? The function and the cell type expression could be very much related intuitively.
Yeah. Yeah, I get your point. Yeah, thanks for the question. Yeah, I think I think that's also the beauty of using LLMs is that it has either its embedded knowledge or its web searching ability that it can find those biological contexts so that it can give a more direct annotation or analysis for the gene set. For example, here I didn't for this task per se, I didn't give it specific biology context of oh this is cancer cell etc. And that was something I was planning to do in the that I acknowledge in the limitation of this approach.
Hmm.
But the beauty of
Hmm.
using LLM is that it is able to do that. Like
Yeah.
if
Yeah.
you're asking specific questions in the context of specific cell types, it it can it should extract information that's specifically related to
Hmm.
that type of information, and
Hmm.
any information that is unrelated and not associated with the cell type should be removed or should not be included. And that's some benchmark or the future evaluation should be built is that if it can filter, it can distinguish the context,
Hmm.
like things in one cell type might
Yeah.
in
Yeah.
a direct in a opposite
Hmm.
direction than the other cell type can it
Yeah.
figure that out. Yeah.
Yeah. So now let's save some time for forward-looking. If you move to your forward-looking slide on the future, the second to last slide, in terms of, yeah, this is yeah very good kind of reference point for us to discuss the future of virtual cell. So one obvious question is do you think your self-supervised learning, the autoencoder, whatever the framework, can also allow you to incorporate longitudinal data like cell cycle progression, even metabolic flux? Do you think you need to move beyond this simple autoencoder infrastructure to some more advanced infrastructure in order to do those multimodal data integration?
Yeah, that is a good question. So from my perspective, I think when I think about this problem is that I think it's depend on the data, but I think what you're saying, like the longitudinal data or cell state, cell cycle information, can I think those type of information can be captured can be captured using the advanced the not advanced the autoencoder, but
Hmm.
I mean that's something that we
Probably
probably
other model like a diffusion model. I don't know because even for imaging data, we know that originally convolutional neural network perform reasonably well. Then vision transformer outperform CNN.
yeah.
So that's one example. Then the regular transformer, it's the natural language transformer, definitely outperform RNN, the recurrent
Yeah.
neural network. So those kind of
Yes.
new architecture, do you think that is needed in order for you to do longitudinal data integration?
So, yes. So from my perspective, is that since the current autoencoder works for this whole pipeline, the first thing to try is to still incorporate the data using the
Mm-hmm.
current
Mm-hmm.
framework. And if we see
Mm-hmm.
things break, then we can bring in
Mm-hmm.
the more advanced model. I think
Yeah.
that's the idea I have is that we
Mm-hmm.
start with things that works already
Yeah.
with the current
Yeah.
space, current framework. Then we add more things on. Yeah.
Yeah. Yeah. So in your method, does this work? Contrastive loss function is very attractive. Simplicity, just a triplet loss. But this triplet loss, due to simplicity, do you think if you want to bring in other data type like subcellular fractionation, proximity labeling, or even in your paper you mentioned high resolution cryo-electron tomography,
Yeah. Yeah.
then can you still take advantage of those simplicity, or you probably need to come up with some new loss function? Or what's your thought along that line?
Yeah, I'm really glad that you asked this question because that's something that the lab is continuing working on is to incorporate more modalities. And actually, recently we have already have developed the model that uh, based on clip model and like advanced the clip model to incorporate more data modality because the challenge of having more data modality with the current method is that right now we are taking into consideration of intersection between the data modalities. And if we adding in more modalities, there is the challenge of we see fewer and fewer intersections because
Yeah.
it's because of technical difficulties and technical differences between all those data modalities. And the new approach that we developed is able to taking the union of between all the data modalities, so it can kind of impute a lot of the missing information from other modalities and able to integrate more than two data types and run the
Mhm.
whole cell map construction pipeline. Yeah.
Yeah, yeah. Another very kind of interesting, probably obvious question, probably I think many of your reviewers also probably ask. You
Mhm.
compare U2OS with HEC293. You see some of the assembly are highly conserved, which probably housekeeping across cell types. Some of them obviously very different because one is osteosarcoma, one is more adrenal gland type of cell types. Then the obvious question for you to build a generalized human cell, virtual cell, how do you computationally handle the cell type specificity? Do you need to have to train different foundation model for different cell types, which is a lot of work? Then you have so many cell types. Or is there a way do you envision that your current embedding space can somehow be predictive on cell state specific variations?
Yeah, so I think the current approach we are taking is that we do have data from other data modalities, and the current computational pipeline that I just described, the unsupervised machine learning, then goes to pan resolution community detection, are kind of are well standardized pipeline, and people just with similar data modalities, you can just run the pipeline and generate the cell map for different cell types, and then directly compare the network based comparison between the cell types. That is the approach, and I think the ultimately we do want to have the model that can you can predict you can from the existing cell type, you can predict what happened if for an unseen cell type or an unseen condition. But
Mm,
yeah, but that might need more information about from the data that if we can generate some cell maps from those data first, then we can
mhm,
further improve the
mhm.
model and framework to do that.
Yeah. Yeah. Final question, wrap whole thing up. Because in your work, you already used for the 3D structure modeling using AlphaFold multimer, which gives you some numerous hydrodimeric complex. Then Of course, we know virtual cell towards the final holy grail of virtual cell. We probably require just more than spatial coordinates. It probably needs some quantitative parameters like reaction rate, enzyme reaction rate, binding affinity, more quantitative, local concentration of certain protein. Do you think your multimodal map can be leveraged or adapted to bring in kinetic parameters to do towards I know you know the simulation is still hard, but do you think your model has the potential to eventually moving towards running some kind of virtual cell simulation by doing kinetically?
Yeah, that is the ultimate goal, right? That is why we are incorporating more cell types, developing dynamics in the cell types. It's trying to understand what things going on, what are things going on in those systematically, and ultimately we can have a model that can just predict what things happened when in terms of perturbation and to move to an actual virtual to move into a virtual cell that we can simulate
Yeah.
experiments, simulate conditions. Yep.
Yeah, more along that line, just quick follow-up, because some people definitely say we should just focus on bacterial cell, single cell organism. One cell is one organism. Or yeast, one cell is one organism, but with organelle. Are you the believer or other collab are believers you should just keep pursuing human cell type of virtual cell? Or do we want to go back to the simple things like a model organism? Can we use your approach to build a bacteria virtual cell? So what's your general thought about choosing the simple cell to model or just go straight to the eukaryotic complex cell?
I see the value of both. Like you first need to understand the simple systems, but I think since we already have the technology, have the data, uh, why not we do things on the human cell? But I can see those routes being pursued in parallel. It's not like one thing or another kind of scenario. It is complementary. Like it gives I believe that they give different
Yeah,
information rather than like conflicting or like same information. So I do see both
yeah,
and both route
yeah,
being really
yeah.
important.
Yeah, later, yeah, we can invite you back during the monthly news discussion. For example, in future, when we talk about companies like Xyra, which will be highly featured in terms of what their virtual cell is doing, or other company in the industrial setting starting to come up with, I think you can be invited back to comment from your perspective when you're analyzing those news. So overall, yeah, really
Yeah,
thanks,
happy.
Kara, for joining us tonight and sharing the exciting work research. And we do appreciate people coming us from all different platforms on YouTube Live. And I just want to have a preview. Our next innovation talk is April 25th. The president of SAPA, Dr. Yi Liu, will give us a deep dive on the history of KRAS inhibition. So our people, we will soon send out the notice. So let's thank Clara again